I have, oh, thank you, Tim. This meeting is being recorded, by the way. I was telling my committee yesterday that I have imagined this actual day many times, and usually when I imagine it, I'm at the Network Science Institute, looking out at an audience of faces of people who, for the last 5.5 years, have taught me so very much. But here we are in this horrid year, having to just trim back so many of the connections in our lives. And so because of that, I'm very grateful that you all came today. And I'm realizing that like one byproduct of this is that having this event be virtual means that lots of different people from different aspects of my life can join. And so people who may not, or who may know me for lots of different reasons, scientifically or personally, just to kind of give a background of what I've actually been doing as a researcher these past several years, is that some of you may know me from some of my work on COVID recently, or about decision-making and experimental design. And in a sense, a broader takeaway of this whole dissertation in general is that all of these things are network science. You get to see a lot of different fields. You get to be sort of just collaborating. And this is actually the most important part. You get to collaborate with such a wide disciplinary range of folks. And my favorite kinds of collaborations are these that just emerge kind of spontaneously, like at a cafe or at a bar. We go back and forth, just scribbling ideas down long to the evening. And kind of fittingly, the origin of most of the ideas in this presentation are from those kinds of events. So this is, I save all of the pub napkins that we draw. If you've ever drawn one of these with me, just going back and forth, talking about ideas. And it just fittingly makes me miss so many of you to not actually be giving this in person. And it makes me miss the really annoying arguments that we would have. So everyone's favorite, or maybe argument to have with a network scientist is usually starts with this, like I'll be like, is network science a science? And then everyone usually in unison and grumpily just shouts all of their answers all at once, because in network science, a very sort of tried and true tradition is arguing about this question for maybe once a year. But what I want to actually do is less jokingly, I guess, present this dissertation in the light that how we answer that question of whether or not network science is a science is going to impact the type and the scope of knowledge that we produce as a field, and it changes what we seek as scientists. And so for this dissertation, I'm going to be casting network science as a science, or at least aspirationally so. So it might not be sort of it might not be the chemistry kind of science that we think of, but in a sense, it has all of the makings for it. And I'm reminded of this really nice Ernst Mayr description of the emergence of biology as a discipline. So you can imagine before biology as we know it exists, there was like botany and medicine and anatomy and all these different spatterings of fields that were not necessarily unified under one roof of quote unquote biology. And what I think that sort of gradual unification of these disparate fields into one biology science has done is that it gives people the sort of intellectual wherewithal to travel from one discipline, or to take insights from one organism. So say I study birds and you study insects. Suddenly now we both study life. And that insights about life in general will apply to both of our organisms. And so what the sort of branding of these are within the domain of biology has done is that it gives us a language to talk to one another. And I think that analogy between biology and network science is useful for our discussions here. For example, the introduction of one really special concept, the network, has had the effect of bringing together otherwise different branches of knowledge. In biology, like a similar unifying concept might be life itself, the notion of life itself, natural selection or genetics. And I think where it differs, and sort of the prospect for us as network scientists comes in this gap, I think, in theory. And so just a quick primer on what theory kind of means to me is that these two quotes sum it up nicely, but it's essentially these objects that point to new directions of scientific discovery, either redefining a subject matter, unifying observations, or in essence, constructing an objectivity through which to view new observations and for designing new experiments. And I'm like 5 minutes into this talk already, and I haven't talked about my dissertation yet. What does this have to do with it? Well, suffice it to say, I want theories of networks. I think one of the big driving principles of me as a researcher is that I'm like, I think, constitutionally drawn towards seeking these types of things out. I want the byproduct of what happens when insights from one domain's networks can inform another science's networks. But before we even have that conversation, I think there's an important step that we need to sort of get on the same page about as a discipline, which is that we need adequate toolkits and benchmarks and standards to be the building blocks of the theories that we construct as network scientists. And so this dissertation is broadly trying to address technical problems in network science that ultimately reveal themselves, or that reveal theoretical challenges about how network science as a field should progress. And so what these 3 chapters of my dissertation, coincidentally, the title of my dissertation, Constructing, Comparing, and Reconstructing Networks, attempt to get at is just like scratch the iceberg of this methodological stopgap that I think will bring the field forward to develop new theories in general. So chapter 1 is primarily with collaborators Eric Hoel, Mike Levin, Ross Grubino, and Anjouman Swine, and Sam Scarpino. And it's taken place over a number of papers the last couple of years. And the key questions that are driving it is about what an informative network actually is and why we might want that. And also, are there questions that we can ask about the right scale to model a system at? If we have informative networks, do we know the right scale to interpret the dynamics from some system? So usually network science is all complicated, and I want to simplify it down to just a single node. This is us. And what I want to drive home is these 2 competing concepts, or maybe drives for what we might want in our complex networks. And it's necessity and sufficiency. So where we are is this node is in the present. To the left is the past, and to the right is the future. And I'm just going to describe some very simple types of relationships that capture these points. So this is an example of a necessary relationship. So B in the past is necessary for A in the present to happen. So you can kind of think of it as a random walker on B with probability 1 traversing to A. Similarly, there is a present to future relationship where A right now is sufficient to bring about C in the future. So there are these 2 competing sides of past and future that we see a lot in the networks that we study. And we can also see more complicated versions of this, where suddenly when there's this new node that can lead to A, B is less necessary, so to speak, than before. Similarly, when A can lead to different nodes, A is less sufficient. That's a wrong use of the word, but for all intents and purposes, it's less sufficient to bring about C from before, and so on. And so networks are noisy and nonlinear and challenging to study. And so the claim of this chapter is that in order to actually understand them, we need informative networks. And this notion of informative is a technical term that I'm going to define using these two competing quantities called degeneracy and determinism. And to still kind of ease this out, I'm going to give you some visual examples. So determinism, I'm going to compare the determinism of these 2 networks. The first one shows a network where if you're a random walker on A, you look at the probability of next states that you might walk to in this network, and you will certainly go to B. So with probability 1, you'll traverse to B. Compare that to this network where a random walker on node A is equally uncertain about its next destinations. And so what we say is this network has higher determinism. And the second quantity, degeneracy, can be sort of best understood as if you're a random walker on B and you look backwards in time and say, where did I come from? You are maximally uncertain about your origin. compare that to this network where no node has any higher certainty or uncertainty about its random walker's previous states than another. So this is maximally indegenerate. So there's all these, this has higher degeneracy, the one on the right. So because complex systems are noisy, nonlinear, challenging to study, and because I said we need informative networks, I now have these two quantities that I'm telling you do capture this idea of informativeness. And so what you might want is, I want to maximize my determinism and I want to minimize my degeneracy. And in more technical terms, this is just basically a difference from the maximum entropy. And what you can do is you can combine these two terms into one single measure. And this is the sort of start of this, chapter. I've with these two terms have defined this effective information. And so it's one measure that captures the extent to which a network describes sufficient and necessary interactions within a system. It's essentially the determinism minus the degeneracy. So I have a new favorite measure. Let's put it on networks and see what happens. So what I'm going to do is just show you a couple of different versions of the same general idea, which is that I'm going to generate a bunch of networks of different sizes, of increasing sizes. And I'm just going to look at how the effective information of those networks change as you increase the size of the network. And the two ensembles I'm looking at are Erdős-Rényi random networks, which, if you're unfamiliar, is parameterized by a single parameter p, which is the density. So it increases in density as p increases. And nonlinear preferential attachment networks, which are characterized by this preferential attachment kernel, which basically makes it more or less star-like as it increases or decreases. So what happens in these two plots, as the colors get darker, the parameters increase. So on the left, you see in Erdős-Rényi networks, denser networks, so higher values of p, have less effective information. Sparser networks have more effective information. This makes sense as you're increasing the size of the network. And interestingly, what happens is that after a certain point, you stop to increase your effective information. So there gets to be a point where the density of your network is constraining the amount of how informative your network can actually be. This kind of makes sense. ER networks are dense ensembles, fine. In the second graph, in the second plot, I'm varying alpha, so I'm making it more or less star-like. And what you see is after alpha equals 1, so after scale-free networks start to emerge, the larger and larger networks you have, the less and less effective information you get with increasing size. So just to kind of say this, to put a point on this, is that the higher density you have, the less determinism, and the more star-like you are, the more degeneracy, so the less effective information. And if you kind of take a step back and think about the networks that you study, whether it's like brain networks or economic networks or social networks, is that they're often characterized by these two kind of competing ideals. So one is you have dense subgraphs of, you have a community structure in networks. So you find these regions of high density. And the other one, you have heterogeneous degree distributions where some people have tons of friends and most people have few friends. And so many networks that we look at actually have these two properties. And what I'm just telling you now is that those properties are not actually contributing to how informative that network is. And this is where the key parts of this chapter come in, which is that there's a way of coarse-graining the microscale properties of this network such that the macroscale network has a higher effective information than the microscale. So what that means is that this is basically a phenomenon called causal emergence, and if the word causal is throwing you off, then you can think of it as like a plausal emergence. I used this joke like a couple of weeks ago, so it's probably not landing that well. But essentially, what it is this. I like to think of it as a smushing of rows and columns in an adjacency matrix, where you have some microscale network. There are two nodes you want to merge together, and you just smush them. It's a linear algebra term. And what that does is it makes a single node, whereas previously on the left side of the plot, there's a microscale network, and on the right side, there's a single node that occupies those two nodes as positions. what it does is it preserves the distribution of the steady state of random walkers on the network. This type of coarse graining is meant to preserve as much as possible about the microscale network, while also just throwing out a bunch of nodes, because those nodes are not actually contributing to the effective information of the network. And so what this is saying is that the network on the right is a more informative scale through which to view the system. So two things happen in causal emergence. I'm just saying, repeating myself again and again. There's an increase in effective information and a decrease in network size. So we can play our favorite game, which is very alpha in nonlinear preferential attachment models, generate a bunch of networks and see what happens. So that's what I'm going to do here. So just a quick reminder, at alpha equals 1, that's a scale-free network start to emerge. So that's maybe something that we want to look out for. And on the left, I'm just going to plot causal emergence, the quantity, which is the difference in effective information between the macroscale representation of the network and its microscale counterpart. And on the right plot, I'm gonna just plot this size ratio. So what fraction of the original network is the macroscale network? And what you see is that these different regimes start to emerge where suddenly lots of causal emergence happens. And around as the network structure becomes more and more heterogeneous in its degree distribution, you get this regime where it is much more informative to cast this network as its macroscale counterpart than it is as a microscale. Similarly, at that point, you get a lot of network compression kind of for free. So you take this much more complicated network structure, and what you get out of it is a coarse-grained description of it. And so, kind of taking a step back, like this plot came out about a year and a half ago, 2 years ago, and it is to this day one of my favorite kind of encapsulations of this phenomenon. And so, what we tried to do is think about in what natural systems might we expect to find this type of phenomenon. And in some sense, biological systems have been long theorized in language that's very similar to this type of language, have been long theorized to have these types of properties. So in particular, what we looked at was this notion of causal emergence in protein interaction networks. And the reason is because there's a lot of good theoretical work kind of building the foundation that suggests we might expect to see noisy microscale interactions leading to emergent macroscale behavior. And sort of coincidentally, there's a really nice data set. So there's this data set of about 1,800 protein interaction networks from Marinka Zitnik's group. And what we essentially use this data set to ask is are there causal emergence differences between prokaryotes and eukaryotes? And for those of you who don't know what that means, I don't particularly either, but a pretty loose characterization is that prokaryotes are thought to be older, are thought to have higher specificity or more like efficient resource use, whereas eukaryotes thought to be younger, are thought to have more noisy interactions, maybe more functional modularity. Some of these kind of buzzwords that ring out like they ought to have higher causal emergence than their corresponding than prokaryotes. And so what we can do is we can play our favorite game, which is just do causal emergence on all of them and look at how much more effective information the macroscales have than their corresponding microscale. On the horizontal axis here is not time, but you can kind of think about it as like where the species is on the tree of life. So what I'm going to show you is that in fact we do find that eukaryotes, eukaryotic protein interaction networks do have higher causal emergence than prokaryotes do. And so this is just sort of one in a now much longer spree of interesting results about thinking about the ways that biological systems might have informative higher scales and how we might be able to capture that with some of these types of information theoretic network scientific methods. So that's chapter 1. Taking a breath. Chapter 2, switching gears, is going to be about comparing networks. So this work is with Harrison Hartle, Stefan McCabe, Alexander Daniels, Guillaume Sanoge, Charles Murphy, and Laurent Hébert-Dufresne, and it recently came out in the Royal Society special issue on a generation of network science. And in essence, we're looking at this question, which is like, you have two networks, like how different are they? How close are they? How similar are they? And can we put a number on that? And the sort of unfortunate answer is that's a really ill-posed question, because if you think of the same question, but for mugs instead of networks, you could say, well, okay, fine, they're different in a lot of ways. Like they're this far apart physically, they are different volumes, maybe their temperatures are different, or maybe their functions differ. And in the same way, you can think about comparing graphs. Like there's a lot of different ways that you can actually quantify the differences between two networks. And there's not before now, I think there hadn't been a great way of actually of performing this comparison tool across all the different ways to actually compare networks. So what we're talking about are these objects called graph distances. A really nice sort of definition of this problem of graph distance was introduced by co-author PhD student Alexander Daniels at the University of Vermont. And you can essentially think of it as a descriptor plus a distance. So the descriptor is something interesting about your network that you want to compare. And then the distance is just the tool you use to give you a number about how different those descriptors are. So we can, for example, a well-known distance is the Hamming distance. So the descriptor is the adjacency matrix, and the distance is the Euclidean distance. Or we might look at the Jensen-Shannon divergence between two networks as degree distributions. We can get a number that way. And they can become more and more kind of complicated or involved. For example, this is my classmate Leo Torres's distance that takes the Earth mover's distance between the eigenvalues of the non-backtracking matrix. And that was introduced because there's strong sort of grounding about why this property in particular is a meaningful one for comparing networks. And in fact, there are dozens of these. Like there are so many graph distances. These are 20 out of many. And what we're trying to do with this paper is essentially make the claim that these tools are really only useful in network science if we understand what they measure and if we can sort of characterize how they differ. And I think a problem that was motivating us was there hadn't really been a good benchmark for this until now. So what we're introducing with this work is this within-ensemble graph distance. So the definition is a mouthful, but we'll walk through it. For a given graph distance measure, the WEGD is the average distance between pairs of graphs with the same parameterization independently sampled from a given ensemble. What does that mean? So, I mean, you can kind of think of it as like, if you're just writing lines of code, like this is just what you would do. So step 0, select your favorite graph distance. I'm gonna pick the portrait divergence for now. Select a graph ensemble of interest. Let's say Watt-Strogatz networks. Parameterize those networks, and then continually sample pairs of graphs from that ensemble, compute the distance between them, do that a ton of times, change your parameterization, and what you have now is this really nice setup for this sort of closed comparison scheme of different distance measures. And the reason that I think this is actually useful is because, or this approach is useful is because random graphs themselves are well studied. There's a lot of ensembles that we know a lot about. And importantly, we can tune them to suit our questions. So we can say, for example, let's say we have GNP, which is a very common random graph model, which is just n nodes and p probability that any pair of nodes share an edge. I can vary p, I can lower it or raise it, and then we can be at a cafe just arguing about each other about what the curve should look like. What if we had the perfect distance measure? What should it look like? What properties would we want it to capture? And then we can get into more arguments and lose more friends. Or we can do a preferential attachment network where we're varying alpha just like we were before, and we can do the same thing, lose more friends. But there's tons of ensembles and there's tons of graph distances. And so the combinatorial problem is quite intense. In the paper, we have this figure or these 6 figures, but I'm not going to show that here. But just to kind of give you a sense of this, and I'm not going to go through all of these, but there's different distances that pick up on different well-known properties of the ensembles in question. And that going into that, if you were to ask us blindly beforehand, we would say that we want that property to be found in our graph distance measure. We want our distance tools to be able to pick up on that because we want them to be useful. And so what this eventually builds to is this big table that's essentially like years of work into a single table. I'm not going to go through this all now, but essentially there's, just to kind of repeat the point, there's a ton of different ensembles, and we know a lot about them. We know important properties about them that are important properties because they correspond to how we use network science in the real world. And so if we're comparing networks using tools that maybe can't pick up the property we want them to pick up, then we're astray as a field. And so knowing this beforehand and having a system set up to actually sort of preempt that and then in turn develop new distance measures as a result, I think is quite useful. The one thing I'm saying, this is kind of a tangent, but the one thing that this also does too is it gets us a notion of a standardized graph distance. This is a pun. I'm not going to explain the pun, but we can call it a meter. So you can imagine you have your network, and you slowly change like one link. Like that network and your original network are pretty close together, right? And so you can imagine changing 50% of your links and then 100% of your links. And suddenly you have this kind of nice notion of standardization, because what you can do is you can use the within ensemble graph distance of your favorite ensemble, so in this case GNP, to actually standardize your graph distance measures such that a standardized graph distance of 1 means that these 2 brain networks that I'm comparing, or these 2 economic networks that I'm comparing, are the same distance away from each other as 2 randomly sampled ER networks. And that may or may not be useful depending on the setting, but at least having that as a benchmark is quite useful, I think, especially in the following chapter. So I'm gonna close this chapter here, but I will say, like, this is, I think, some of the most fascinating and simultaneously boring science I've ever done, which is just like sample a ton of networks and measure their distances and see what happens. And I feel like a kind of kid just like playing with code. So and there's also, this is very much unfinished work that I think there's a ton of things to do here. So that's it. Chapter 3 is related but different. So this is about reconstructing networks, and this is largely the, maybe not culmination, but it originated as this large international collabathon that we hosted out of Northeastern now, oh my God, almost 2 years ago, where we got a bunch of people together to implement different techniques for reconstructing networks and measuring graph distances. And I think 2 broad things came out of this, and there's sort of 2 halves to this chapter. One of them is the technical contribution. So we made a software package that a lot of that I'm quite fond of. And the other one is a more theoretical approach. So I'll get to the technical bit first. The problem is, or maybe not the problem, just like what we're faced with is that, so this is recordings of neurons from a mouse's visual cortex, and like, we're network scientists, so obviously there's nodes here and we can kind of squint and see some links, so let's just draw them. And there we go, we got a network. Easy, right? And so this general structure of this problem is actually quite common across networks. It's not just neuroscience. So a complex system will produce some interesting behavior. We will put sensors in that system to record that behavior. And then what we'll try to do is we'll try to use the time series of data that gets recorded to understand that system. So the general structure of the problem is like, there's some quote unquote true network structure. We can talk about that later. We have some time series data that comes from it. And from that time series data, we try to approximate the true network structure using a bunch of reconstruction techniques. And if this sounds like a similar framing to the previous chapter, that's because it is. There's a ton of different ways to do this. And in a sense, how do we compare the reconstruction techniques here? Well, You can open up your Python right now and pip install netrd 2.2 or 0.2.2. It's a cool library. Lots of people have recently downloaded it. And the basic pipeline that it lets you do is you can sample a network using network X. You can generate some observable dynamics using some of the dynamics that are built into this package. And from the time series that gets generated from those dynamics, you can use a number of different reconstruction techniques to try to approximate the original network structure. And then once you have that, you can use a bunch of different comparison tools to actually tell you how far either from the original network structure you are, or you can study the reconstructions themselves as objects themselves. And you can say, these ones did better on this dynamics, these ones did better on this network structure. The combinatorial adventure awaits. So that was a brief intro to the technical side of this. The theoretical contribution is a little bit more, this is a good one to kind of end on because it's a little bit exhausting. But it's an ensemble, it's a framework that I'm going to try to introduce in the following way. So new framing. Consider your brain. Also, apologize for talking about brain so much. This isn't meant to be about brains or neuroscience in general. This is just a good example, I think, or a good case study. So consider your brain, your friend's brain, and their brains, and so on. And suddenly you have this space of all possible human brains. So you can kind of pick out where you are in this. And correspondingly, there's also a space of all possible human brain networks, so all the ways that these neurons can connect. And this might seem like a big space, and it's going to get even larger because there's also the space of all possible human brain dynamics. So if you think about all the possibilities, all the different ways that this could combine to create you or us, that seems massive. But if you think about it, it's really just a teeny, minuscule fraction of all of the possible networks and all of the possible dynamics. So in that sense, it's quite a nice little encapsulation of an ensemble of networks. And what I'm going to try to introduce here is this GDR ensemble that it just rolls off the tongue. So the GDR ensemble has three parts: G and D and R. And the G is essentially some graph ensemble that you can randomly generate instances of new graphs from. So this could be, as in chapter 2, we selected a number of different ensembles to compare distances between them. On those networks, we run some dynamical process that creates a time series of data, D, or dynamics, D for dynamics, for which we reconstruct or attempt to reconstruct that original network structure. But what we have here is a kind of sort of closed setting, where if you zoom out for a second again, and you think about the GDR ensemble in real systems, imagine that setting we were just in, where I was talking about the space of all possible human brains. Those brains produce some space of all possible human brain dynamics, which we can get at through a number of different reconstruction techniques. And importantly, where we put emphasis on like theoretical advances and algorithmic advances, this quite clearly kind of delineates the different roles of each. For example, neuroscientists for years have been trying to characterize what they think is like a most appropriate way of describing brain network structure at different scales. Obviously, I'm grossly sort of generalizing this, but we have a good theoretical infrastructure for actually saying we think human brain networks look like this. We also have pretty good theoretical tools for describing brain network dynamics. So like, and then the reconstruction techniques that we have can be iterated upon and improved to suit the actual best in practice, best in show tools that we have for actually getting at G and D here. And what we can do with this is that when we actually use these networks to intervene in real systems, what we now have is a much better closed environment to do some sort of perturbational testing. So we can say what would happen if this brain area were lesioned, or what would happen if this sector of an economy had some sort of intervention into it. Because we have put and isolated our theoretical work into well-defining G and well-defining D, we can then focus our energy on getting good reconstruction techniques that advance theory. I'm going to close this example with let's say someone told me the true brain network structure. So let's say G is nonlinear preferential attachment networks, because it's getting old. D is branching process dynamics. So this is God saying this is the two types of G's and D's that we're going to work with. What we can do is then measure distances among those reconstructed networks. This is my favorite game. I don't know if you've realized this. But for G, there's different ways that we can parameterize. These are all different brains, by the way. You can see yourself somewhere in there. For D, the structure of those networks actually inform the types of time series that we'll extract from them. And as such, what we can do now is we can pick a few of our favorite reconstruction techniques and measure sort of standardized graph distances between them. Only the standardization here is not GNP, it's this G. It's these brains as our standardization. So what is this figure? This figure is now, again, on the horizontal axis, we're varying alpha. It's a favorite game. Each subplot is a different graph distance that we're going to measure the distance between the reconstructed networks and the original ground truth networks. And what I'm going to do is I'm just isolating two different reconstruction techniques. One of them is via a Granger causality method. The other one is via minimizing a free energy. And they both reconstruct some network. Are those networks similar to A, each other, and B, the original networks? That's an open question. So what we can do is we can look and say, in the degree divergence, which is just a certain distance, these two curves look kind of similar. They minimize out at a similar point. So that means the reconstructions under this lens are similarly dissimilar to the ground truth networks, in that A, neither of these curves go below this this dashed black line. So neither of them are closer to the original ground truth network than randomly generated, in this case, preferential attachments are from one another. We can look at a different example, another distance called delta con, where this is much closer to that 1 threshold. But in one case, the free energy approach stays under 1 this whole time and minimizes at about alpha equals 1, whereas this Granger causality minimizes at 2, and importantly, is not below this distance of 1 threshold at low values of alpha. Where this gets, I think, especially interesting, and sort of the whole proof of principle of this long spiel that I've been on, is where they start to disagree, which is as alpha increases beyond 1, not only do they diverge, the distances between the reconstructed Granger causality networks and the reconstructed free energy minimization networks, not only do they diverge, but in fact, one of the techniques starts to generate networks that are farther away than two randomly sampled preferential attachment networks. And so this type of, I mean, I think it is a kind of insight to gain from because this framework has been introduced, is that you can imagine a different scenario where it's not nonlinear preferential attachment networks that are defining your like true brain networks. It's years and years of neuroscientists saying, no, I think networks have this type of structure. I think networks have this type of structure. And this convergence of theoretical work generating different ensembles of networks that like lead you to a point where you can say, actually, there's a specific point where these two reconstruction techniques disagree. And this is the domain where experimentation can actually resolve some of these disagreements. I'm just going to skip over this Ibsen-Mikhailov, which is similar in interesting ways. So that was a introduction to, I think, what is a much larger iceberg of this framework for thinking about reconstructed networks. And in general, this idea that I mean, if you think back to the beginning of the talk, like sort of, this is getting to the end of the talk, so I'm trying to think about how to close this. But one way of reminding you what I'm doing here is that I really want to think about theories of networks. And as I said before, in order to even consider theories of networks, we need to be sure we have this adequate toolkit or these benchmarks or some sort of standards as the building blocks of our theories. I mean, I'm reminded especially of this really nice quote from Hasuk Chang's Inventing Temperature, where he says the scientific study of heat started with the invention of the thermometer, and that this is a well-worn cliche, but the comparison here is useful and highlights a couple of similarities between the study of networks, where the thermometer is a useful measurement tool. Similarly, data scraping or survey methods or reconstruction techniques, et cetera, are useful tools for measuring networks in different systems. So in this example, a key difference is that today's thermometers would not have emerged without theory. In particular, the theory that there are approximate fixed points in the phase space of water. And as far as I know, this analogy stops here, because I'm not sure there are similar sort of theoretical objects in network science. And in a sense, that's precisely the point. Because there's a sense to which we can consider every network we study, every network we use in our day-to-day lives as network scientists to be little hypotheses about the systems in question. So that is, it is my best guess about the structure of interactions in some complex system, and implicitly encodes like what I think would happen if I were to intervene in the system. So if I were to add a new friend connection here, or if I were to lesion this brain region, or remove this actor from the economy. And so the question that I want to sort of encourage us as a field to ask, and that I've been trying to ask for several years now, is that if this network is my hypothesis, then what the heck is my theory? And so moving forward over the next couple of years, I want to really drill down on this idea. And surely there's people who have thought of this already and deeply, and it's going to be I think an exciting like learning process to actually get through that. So it's 1:45. If you'll indulge me 5 minutes, I would like to kind of just.
That is, I have to call you on time because
okay.
Of all the committee, and I don't know if anyone in the committee has other. The scheduling is complex, you know. We are not in person,
for sure.
I'm so please try to go to the conclusion.
I mean, yeah. in that case, there's just like I have been so unspeakably fortunate to get my PhD at the Network Science Institute, because what it means is like you hear directly from the source of so many of these really interesting, like inspiring when you're a junior researcher in this field, insights that I mean to like I wish it were under different circumstances, but to be here today is just such a immensely proud. I'm so very, very grateful to have gotten my PhD around these amazing people. So thank you very much for coming today, and I really appreciate any questions you have, and thanks.
So I clap hands. It's always a little bit awkward on Zoom, but it was a great presentation, and I think again it's how to say, all the narrative that you have built around your work is exemplary, and it was a great presentation, and I open the floor to questions, please. Is there any question from the audience? You can how to say, raise your hand or just unmute yourself and ask the question at the moment. Please don't be shy. This is your last chance to grill him and let him suffer a bit. So please. Instead, a lot of congratulations, you know?
Yeah, I saw hands being raised. Oh, so Lionel has his hand raised.
Okay, let me see. There is Lionel Toba, please. Can you unmute yourself and ask the question?
Hey, yes, can you hear me?
Yep.
Yes.
All right. Thank you. Thank Brennan. That was good. So just something regarding the comparing parts of the presentation. So comparing network. So on the practical sense of it, why the need of comparing networks. So say you have, or if you want from a modeling perspective, right, you have two different systems, you have two different networks or two different modeling. Why not focus on studying these specific or the specifics of those networks and instead compare them? Like, how would they help in solving issues in those specific networks?
Yeah, so I think it's a good question. It's also, like, in some sense, a question that we had to write and rewrite in the introduction to this paper, because you have to kind of convince yourself that it's important to compare networks. I think two broad things. One of them is so you may be studying a single system, but if that system changes over time, how do you actually know, or how can you characterize its evolution? That's one side of it. The other side of it is, and this, I guess, more specifically to the within-ensemble graph distance idea, is that having sort of benchmarks, or at least like starting lines to compare things to, is, I guess, fundamental in not just network science, but in science in general. So having the ability to actually characterize how far away something is from our expected result is, I think, it exists in many ways in network science. So we have null models, or we have different null models. But this is another way at that question of just saying, I expected something to look like this. The network that I actually have looks like this. What's the gap there? And in particular, can I define a set of tools that specify what that gap looks like? So is this graph distance, or is the gap between what I expect and what I have, does it differ along these axes? Does it differ along some other axis, etc.? Hiroki also has.
Yeah, and then there is Hiroki was another question. Yeah.
Thank you very much, Brennan. This is fantastic talk. So this gives me the challenge. How can I make my students to this level? But anyway, so my question is this. So let's imagine that you pass this defense, you become a Dr. Klein, okay? And then you go out and then the people actually approach you. Hey, Dr. Klein, you are the expert of graph distance and graph reconstruction. People out there, they just want to know the answer. So when they say, tell us, which method we should use for this particular data set, what would be your response to that kind of consumer mindset type of question?
It's a good question. It's also, I think, so the short answer is I don't know. The longer answer is I have some thoughts. So let's take the case of graph distance. And this is something that I sort of squirm when I hear myself say this. But what I would do is I would use all of the graph distances, just kind of look at how they all, what numbers they get. So I have two networks, or maybe I have a class of networks that are control brains and a class of networks that are brains that have maybe been diagnosed with schizophrenia. I want to know how different they are. And I want to use the right tool to characterize how different they are. What I would do is just use all of them. Mm-hmm. The reason for that is because if you actually know the mechanics of the distance measures that you're using, you know what's different about the networks. So if under the degree divergence, let's say, these two systems are the same, then they don't differ along the meaningful axis of their degree distribution. If, on the other hand, the spectrum of their non-backtracking eigenvalues is wildly different, then you know something specific about, this is like trivial, you know why they're different. But then, and just to kind of close this off, is that like you as the domain expert who is using this distance, the task now is on you to say, okay, I know these two sort of categorically different groups. What is it about the pathologies of this group, or the sort of controlness of this group, that would bring about differences in their non-backtracking eigenvalues, or their quantum density matrices, or whatever? And so then it reframes the role of the theoretician, the scientist, in I think a useful way.
Yep. Thank you very much.
Yep. Matt.
Matt Simonson. Yeah.
Hi. Amazing talk, Brennan. Really impressive. I was wondering about these really cool ensemble approaches you have. Could you use an empirical ensemble? So let's say you measured, 10,000 different rat brains of one kind of rat, and then 10,000 rat brains, another kind of rat. Could you use an ensemble approach to compare how similar these two species brains are on average? So rather than using the combinatorial way to approach an ensemble, just having an ensemble that is a bunch of specimens that you're comparing.
So I am super fond of that idea. I know there's currently being work that touches on kind of exactly that. Just a quick, this isn't a sufficient slide. It wasn't a full enough slide to actually put in the presentation, but I sort of did that exact example with healthy brains and brains that have been, or a data set of like 150 brains. Some of them had diagnoses of schizophrenia, others were control brains. The within ensemble graph distance under this non-backtracking eigenvalue distance of the healthy brains has its own characteristic distribution, and as such has its own sort of characteristic shape. And so what you can expect is that, or what you can maybe claim, is that this approximates this space of all human brains, let's say. And so it has like, it's this multidimensional object, it has wobbles in all of these different ways, and you can kind of measure those wobbles in a meaningful way if you have the right tools to measure it. What you can then do is say, I know that this other group is different or might be different. And what I want to do is measure some properties of the distances in that group. Yeah, short answer is yes. I don't know if that actually needed that big of an explanation, but.
Okay, there is another question by William Brush.
Billy.
Please.
Hey, Brennan, can you hear me?
I can.
Excellent. Thanks so much for the great talk. That was a delight.
Thank you.
So as this is probably gonna be a naive question as someone who doesn't think about this field at all, but I wanted to ask you a question about your section 2 or chapter 2. In your talk, the main point I got is that you should think carefully about what graph distances you want to use depending on what properties of the network you're interested in. And yeah, stop me if that's not fair. But
that's correct.
So it makes sense to me that the properties you're interested in should be dependent on what questions you're asking about a network or complex system. And the questions you can ask about a network all come down to what are the nodes and weights that you can assign to edges between different nodes. Is there a way to think about the space of questions you can ask about, some high dimensional space, and then try and come up with some link between types of questions you might be asking and types of graph distances you want to use? And in some way, yeah, so that's kind of the part A of the question. And part B is you're saying that you answered one question by saying that you should just use all graph distances to try and inform your answer. Well, what if in this large space of questions you can ask, all the graph distances, maybe except for one, are kind of all clustered, and one is like very far away from these distance measures? You'd expect a lot of them to agree and maybe the other one to stand apart, but maybe you want to kind of like sample the question space more evenly. Is this anything you've ever thought about? Or?
Yeah, I mean, the line of questioning is like, is terrific because it touches on several different important parts of what I want out of a network science, which is that, number one, like, I think hidden in those questions is that it's us putting the network on this system. We are saying this is a node. We are either measuring or assigning weights between nodes. And in that world, we are constrained to only ask questions of those types of networks. And so that this question space of the sort of network science then can be specified. What I think theoretical work will like help pin down is how to carve that in a way that we get the like, not like purest network science, but just like most firm foundation to actually to ask these questions. That's a sort of general point. The point about the question B, where let's say I have 10,000 graphs and they're all the same except for under net simile, this distance. Like what why, and also like, do I care? And also, what does it inform about network science? One of the things that I think that poses is like, certainly there's some differences between these networks, and certainly you can imagine if you didn't have that net simile distance, you would say these networks are all the same, quote unquote, or that they are all, and I actually think you would be correct in saying that, because until we actually have measurement tools that can properly distinguish, until we have that like outlier distance measurement, we don't actually, we literally cannot observe the meaningful differences in these networks. The same sort of problem exists in the reconstruction space, although I'm worried a bit of time. So I think I'll just stop the answer there.
Thank you.
Yeah, so now in the interest of time, I will have to cut short the following two questions, which are the last one. So Carolina, please, your question, and Brennan, please, a short answer, because we are running out of time.
Alrighty, I'll be concise. Would you say that the GDR framework might generalize to network dynamics that don't exactly produce time series?
Yeah, I thought about this. I don't think I'm good enough at dynamics to know the answer to that. So, or at least like people deal with like so like sequences of graphs maybe, or paths through networks. I think so, probably. I haven't thought enough about it to actually like be able to quantify it. But I think it's a great and also immediate next question. And Alice, we're on the SINS Slack channel together. So if you would like to ask a question,
Alice,
I'm happy
please.
to.
So, Alice, do you have, can you unmute yourself, or you have,
Thank you, Brennan. This was excellent.
Yes, great.
So, if I still may, I might ask my question, which is about causal emergence. So that started out with some sort of random walks on networks. Now, not everything that happens on networks can be accurately described by a random walk. Is there a way to extend causal emergence in a way that you can account for different types of dynamics? And if you can be really short about it, what I got away from your second and third chapter of your presentation was that there's a huge promise in shifting from looking at single networks to looking at graph ensembles. So when you come from a data driven field, when do I know that I have collected enough networks that I can call them an ensemble and start using the tools that you proposed today?
Ooh, I'm going to answer the second one first, because that I think it's a fantastic question. I have no idea the answer. That's my answer. But like in a sense, there's a data scientific way of approaching this, of saying like it's essentially like a power analysis of saying I have enough subject in my experiment to know that the effect size that I'm looking at is actually significant, or that the sort of distribution of the graphs that I'm looking at is stable as I add more and more new networks in, that maybe I have something that I can squint my eyes and say, yeah, it's on an ensemble. But take that with a massive grain of salt, because I don't know. The first question is something that I've wanted to do forever, and I would love to have collaborators to do this, but essentially if you can have a transition probability matrix, no matter what type of like dynamics you're actually using, you can still get at some notion of causal emergence. All I need to know is what's the probability that a next state happens. If I don't, I can also have a sort of a distribution of probabilities, although that would get a little bit messier. It's something that I would love to do, because I don't know any really random walker kind of systems. But as an initial framework, I think we stuck with random walkers because it was much easier.
Okay, I have to unfortunately stop the question and answer time. As always with Brennan, you know, when he gives talk, then there are thousands of questions afterward, and that's a very good sign. He speaks about how much his thinking is provocative and how to say, creative. Now, you know, let's thank again all the attendees to this talk. He had at a certain point more than 70 attendees. So thank you very much, with a lot of guests from all over the world. So thanks a lot. And I would again applaud the hard work of Brennan. And now we have to move in the torture chamber with the committee. So Brennan, I don't know if you organize anything in terms of breakout room or, and you want to have your friends stay here and, or how we play.
I can try to do a breakout room. My brain is a little bit slow right now, so I'm going to assign the committee and myself to a breakout room.
Yeah.
Right now. So Alex and who else wants to come?
I'm also very impressed that you had Albus Dumbledore as a guest to your talk.
Yeah.
Really, there are, you know, really important people here. So
just trying to get that magical experience. And Laurent. Okay. Open room. See you on the other side. Bye.
Bye.